Today, I'm going to be speaking to you about the bacterium Yersinia pestis and the disease that it causes, more commonly known as the plague. My name is Josiah Weimer. This is my medical microbiology. So this brings us to our introduction. What is Yersinia pestis? And simply put, Yersinia pestis is the pathogen that is responsible for multiple plagues. These plagues include the Black Death, the Great Plague of London, and also a plague in Hong Kong, which we're going to speak about in a second. While this plague was more prominent in the past, it is still an issue today, causing over 3,000 cases between 2010 and 2015 alone. This bacterium was discovered by the Swiss scientist Alexander Yersin, and Dr. Yersin worked in Asia during the Hong Kong plague, whenever that bacterium was very prominent. He isolated the bacterium and noticed rodent die-off from the plague, which was a really big step in figuring out how the disease was transmitted and where it came from. In this presentation, I'm going to outline the following things. First, I'm going to outline the structure and the genetics of Yersinia pestis. I'm also going to identify the pathogenesis and the epidemiology, essentially how the disease spreads throughout the body and how it is transmitted from person to person. And finally, I'm going to cover the detection, the treatment, and the prevention of plague, the disease caused by Yersinia pestis. This brings us to our classification of the bacterium. Yersinia pestis belongs to the domain of bacteria, the phylum Proteobacteria, the class of gamma proteobacteria, the order of Enterobacteriales, the family of Yersiniaceae, genus of Yersinia, and the species of Yersinia pestis. The classification is important because it identifies which bacteria are related to each other and helps us group them based on genotype and phenotype. This brings us to one of the most important concepts of bacteria, which is the structure. And Yersinia pestis is a rod-shaped gram-negative bacterium. So that means it's a bacillus bacterium. So it's rod-shaped, and gram-negative indicates the nature of the cell wall and cell membrane. Essentially, a gram-negative cell does not have a thick layer of peptidoglycan, similar to its gram-positive counterpart. Instead, it contains chemicals known as lipopolysaccharides in the cell wall. And if you break down the term lipopolysaccharide, you can see that it consists of a lipid, fat, lipo, and a polysaccharide, which is a sugar it's a complex sugar with multiple sugars linked together. And this is a major pathogenicity factor in the bacterium. And we're going to speak about that in a little bit. Another characteristic of gram-negative bacterium with lipopolysaccharides is that they have an inner core, and this is referring to Yersinia pestis. The Yersinia pestis is an enterobacterium, and that inner core can be seen here in this diagram right here. You have your polysaccharides, which is a bunch of sugars kind of linked together to make a more complex structure, and then you have lipid A. So this inner core is kind of referring to this whole thing right here, especially the connection between lipid A and the core. And this inner core is acylated by glucosamine substituents. So you're gonna see just a bunch of branches connected to this lipid. And those branches are very important, and I'm going to get to that on our next slide. This is just kind of the introduction here. And it's important to know that the Yersinia pestis undergoes a similar synthesis to E. coli. Since they're related, and E. coli has been so extensively studied, we can kind of compare those synthesis processes. And there are two main components of lipopolysaccharides in Yersinia pestis. We have the lipid A and the oligosaccharide. And that lipid A is responsible for anchoring the lipopolysaccharide to the cell membrane, but it's also important for how the body reacts to the bacterium once it's in the body. And then we have the oligosaccharide chain, and that's oriented towards the outer department or environment. So if you imagine your bacterium right here, that lipid A is going to connect it to the cell membrane. So if you have your bacterium right here, that would be kind of sticking out like a spike. The structure of lipopolysaccharide in the bacterium of Yersinia pestis depends on factors such as temperature, which is very important since Yersinia pestis is almost exclusively transmitted to warm-blooded creatures. Lipopolysaccharides, they'll form varying degrees of acylation based on temperature. And again, that acylation is just the attaching of substituents onto the original structure. And it's important to note here that lipopolysaccharide does not acylate easily in high temperatures. So in this diagram right here, on this side, we have, on this upper left corner, we have a lipopolysaccharide that was made in a Yersinia pestis bacterium that was exposed to heats around 37 degrees Celsius. Now, as you recall, the 37 degrees Celsius is the normal temperature of humans. So this is really important right here. You can see that there's not a high level of acylation. In B, this one is at a much cooler temperature, around 6 degrees Celsius, in fact. And we're going to go into why these acyl groups are important. The reason why they're so important is because we have these things in our bodies called toll-like receptors. And these toll-like receptors are important for recognizing antigens, and they're going to create a cascade of signals. And these signals are going to create an immune response. It's going to create inflammation. It's going to recruit macrophages and neutrophils, and that's going to help neutralize the bacterial threat. But the problem with the low acylation, ones commonly found in Yersinia pestis, is that the toll-like receptors just couldn't recognize these. So essentially the bacteria was able to bypass the immune system, and it prevents the production of TNF-alpha, tumor necrosis factor alpha, which is really important in the immune detection and subsequent immune response. And then another important thing about the high temperature lipopolysaccharide that scientists found was that these lipopolysaccharides could survive in a flea's digestive tract. And this was important because this was the form of lipopolysaccharide that would make the bacterium especially virulent. And then it was also shown that low acyl forms of LPS were not shown to significantly affect the longevity of infected rodents, so the rodents that were bitten by those infected fleas. And this is important because essentially that ensures that the dangerous bacteria could remain alive until injected into a warm-blooded host. Another important characteristic of bacterial genetics is the evolution. And bacterial genetics are central to a bacteria's abilities to survive in its environment. And the evolution allows cells to optimize survival traits to kind of get rid of traits that are not doing them any good. And a lot of times these traits are passed through what are known as plasmids. And plasmids, they're kind of outside the original bacterial chromosome, but they can be integrated into the genome and allow for bacteria to be favored and selected for in more extreme circumstances. And these plasmids are genetic material that can easily transfer between cells. It's important to note the Yersinia pestis strain originated from the Yersinia pseudotuberculosis, and that's just an older enterobacterium. But as we're going to see in a second, this bacterium is going to pick up traits that are going to help it survive, replicate, and spread to other hosts. So, in order to survive and better adapt to its environment, Yersinia pestis gained two very well-known plasmids, and these plasmids are PMT1 and PPCP1. And we're going to go over why these are important. PMT1 is responsible for the first step in the transmission of Yersinia pestis. And essentially what this plasmid does is it allows the bacteria to survive in the digestive tract of the flea instead of being broken down by digestive enzymes. And this is important because if that bacteria was broken down, it wouldn't last it wouldn't last in the flea, and it wouldn't be spread at all. And another important thing is it induces septicemia in rodents, which allows the infection to spread throughout the blood, and then it allows the tissue to become contaminated and helps the bacterium spread the illness a lot better. The PPCP1 is also important because it increases the virulence of the pathogen. And three notable genes found on PPCP1 are the genes PLA, PST, and PIM. And we're going to go over this right now. So PST is known for encoding pesticin, and this pesticin is going to actually reduce the competition from other bacterial cells for resources by breaking down their cell walls and just rendering them useless. And then PIM is also going to help the cell survive by increasing Yersinia pestis' self-immunity to pesticin. This brings us to PLA. And PLA is mostly important for after the infection, after that bacterium reaches that higher temperature in its warm-blooded host. And after the initial infection, the PLA is going to be upregulated because that optimal temperature is 37 degrees, so that's just going to kick up its production really high. And what this gene does is it produces proteins that break down the host's fibrin. And that fibrin kind of acts as a protective measure to contain the infection. When that fibrin is down, kind of like a dam, the water is just going to spill out. And in the same way, that bacteria is now free to spread throughout the body. Another important note is that Yersinia pestis flourishes when the host dies. And the reason for this is whenever our body decays, bacteria start to ferment our different chemicals in the body, and when that fermentation takes place, the pH lowers. And these PST and PIM genes operate the best at low pH, typically in between 4 and 5 pH. And this allows the Yersinia pestis bacteria to grow unhindered, especially since it's reduced the competition from other cells. And other cells typically do not work as well in this acidic condition. This brings us to our pathogenesis of Yersinia pestis. So once Yersinia pestis enters the body, it typically manifests as an incubation period of 1 to 8 days. So in more severe cases such as pneumonic plague, what's going to happen is that Yersinia pestis is going to be about 1 day. And then with bubonic plague, it takes a little bit longer. But the bacterium relies on a type 3 secretion apparatus to transmit effectors into cells. And essentially this type 3 secretion disarms the innate immune system by interfering with cell signaling. And it allows that disease to continue to develop. Remember how whenever we were talking about the structure, the lipopolysaccharide was able to evade detection by the innate immune system, those toll-like receptors. Another important factor is the V antigen, and this one is integral to immune suppression. So rather than just kind of bypassing the system, this one actively suppresses the immune system. It binds to negative regulatory proteins found on the cells, and it actually starts to perforate the cell, and it pokes holes in them and allows the effector proteins to get in there into the cell cytosol and interfere with the protein. Another important factor is Yersinia bactin, also known as YBT. This is known as a siderophore, and the siderophores are important because they have a very high affinity for iron. And this iron is important because it allows DNA, it provides DNA replication, it provides many different functions. It allows blood to carry oxygen, so it has so many different functions in our body. But what this Yersinia bactin does is it takes iron molecules from binding proteins such as transferrins and lactoferrins, and it allows the Yersinia pestis to multiply and cause widespread infection. And for this reason, it's instrumental to the virulence of bubonic plague and pneumonic plague. So since we kind of covered pathogenesis of Yersinia pestis at a more molecular level, we're going to take a look at our symptoms. And the plague begins most commonly in the lymphatic system, and this is known as the bubonic plague. It's the most common form. So whenever the bacterium enters the body, it goes straight to the lymphatic system and lymph nodes and causes these large swellings known as buboes. And these swellings are very common in the axillary and inguinal regions. So that's kind of your first tip. Whenever you see those swellings, you can suspect that Yersinia pestis is at the core. Additionally, fevers, chills, and weakness are also common. And then, if this infection remains untreated, it can also spread to the bloodstream. And this is known as a condition called septicemia. So it's a systematic infection of the bloodstream. And in this continuation of the illness, digital necrosis, so in the fingers, the toes, and the nose, it's very common to see. Necrosis in the death of cells, gangrene, and those cells are typically not able to be saved, so amputation is very common. And then the most severe complication is known as pneumonic plague, and this is whenever the bacterium reaches the lungs and begins infecting cells. Whenever this happens, you can see fever, headache, weakness, pneumonia, and bloody sputum. And this is basically the only complication of plague where it's spread from human to human. Typically, it is a zoonotic infection. And this brings us to our epidemiology, in which we learn how the disease is spread and what we can do to prevent it. So the plague is a zoonotic disease, and we have fleas such as X-Chiopus, which is also known as the oriental rat flea. That's the main vector, and these fleas obtain the Yersinia pestis from the infected animal. Since that Yersinia pestis has the plasmid that we talked about earlier in our presentation, the PMT1. This allows the bacteria to remain alive in the digestive tract, and whenever the flea bites another animal, like a small mammal, a rodent, it's going to regurgitate that bacterium back into the bloodstream, transmitting the infection. Additionally, plague can be contracted by contaminated objects, and this includes body fluids of another person or animal, or contact with infected animals, and in even some cases, eating the meat of an infected animal. And the last one is infected droplets, and that's seen within pneumonic plague. And Yersinia pestis is also seen to spread the most easily in rural areas with large rodent populations. So right now, it's very common in Africa, which has a large wild rodent population, with secondary spots being in Eastern Asia, with high density population and also rural areas, and then also in remote areas of South America. In order to contain this disease, isolation of sick individuals, surveillance, watching them and monitoring their progress as they're being treated, and disinfecting any surfaces are paramount to containment. And the first step in treatment of Yersinia pestis is identification and diagnosis. And this can be done through detecting signs and symptoms, kind of like how we discussed in the last few slides. There's also PCR detection of F1 antigen from blood, sputum, or the liquid from the bubonic swellings, the buboes. And then sampling and subsequent culturing through McConkey agars, blood agars. All of those, even phage treatments, can help detect that F1 antigen, which is key to detecting Yersinia pestis. Early recognition is also crucial for prognosis. So essentially, the earlier the better in terms of recognizing and defeating the disease. Even a 24-hour delay can reduce the likelihood of the disease working. The standard treatment of Yersinia pestis is antibiotic treatment, and this is most commonly streptomycin or gentamicin for adults. And typically children can receive these antibiotics as well, but they usually get them in smaller doses. And with antibiotic treatment, plague mortality rate is now 8 to 10%. So that's a 90 to 92% success rate, which is much better than leaving it untreated. So this brings us to our conclusion. Although bacteria such as Yersinia pestis and their associated illnesses have existed since the beginning of time, the development of modern medicine has allowed societies to better weather these storms. By gaining a deeper understanding of the structure and functions of pathogens, researchers can develop effective methods to increase the quality of life of the societies that they live in. And the thing I kind of want to leave you with is this. We have been given the resources and opportunities to increase the depth and breadth of our knowledge. How will we use this gift? That brings us to the end of our presentation. Thank you guys and have a great night.